My name's Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group. Welcome to our webinar for June. Today our speaker is Tatyana Dobreva, the Chief Executive Officer and co-founder of ImYoo. And Tatyana is going to be talking to us about patient sampling of capillary blood provides unique biological insights into autoimmune flares. But before we start, I'd just like to briefly thank all our partners and sponsors, without whose support we would not be able to bring you these webinars. This webinar is being recorded, and in a few days or a week will be available to stream for free on demand. Just taking you through a little bit of the agenda. Again, I'm Neil Spooner. Then Tatyana will be speaking to us. There should be an opportunity for questions and answers. If you could please submit your questions through the chat function, and I will then be able to read those out to Tatyana, and I'll come back and wrap up with everyone at the end. So, no further ado, I'd like to hand over to Tatyana. Welcome to the PCSIG webinars, Tatyana, and thank you so much for sharing with us. And I'll hand over to you.
Thank you, Neil, and thank you for everyone for being here. I know it's early morning for some of you. All right, I'll go ahead and share my screen. Okay, can someone confirm that you guys can see my screen?
Yep, can see it, Tatyana.
Okay. All right, awesome. Okay. Well, hi everyone. I'm Tatyana. I'm the CEO and co-founder of ImYoo, and I'm excited to tell you about what we're building. So ImYoo is creating a patient-driven biodiscovery platform, and what that means is we are changing the relationship between patients and researchers. We want patients to be active participants in the research that's being done to develop better medicines for them. I'm an autoimmune patient myself, and it's actually a primary reason of why I started ImYoo. I had a long diagnostic journey myself, and it took a while to find the right treatment. And I know that the tools and the data that we can collect is out there to make more effective medicines for everyone. All right, let me give you a little bit of context before I dig into our autoimmune flare study. So just as you guys know, and we all experience with COVID, everyone has a different immune system. Everyone responds to health adversities differently, and every person has a very rich and unique molecular journey. However, when we study diseases in our traditional settings, we only see a small slice of time, and not just a small slice of time, but only the most dominant signals. So what we're doing at ImYoo is thinking about what if we could see the whole picture across genes, across cells, and across time. And not just from some people who are situated in urban centers, but from people everywhere. So what we're doing is enabling patients to collect single-cell molecular data from home. So patients self-collect blood using the device that Emily has here on her arm. They can do it at home. They can do it when it's best for them and during their disease activity instead of following a study schedule. So our agenda will be to talk about, so how does going decentralized affect the kind of data we're collecting? What new molecular targets can we detect during disease activity specifically? And can molecular signatures be used to stratify patients within an indication? So before going there, it would be good to understand what is ImYoo's platform. We call it the REACH platform, where we go directly from patients all the way to insights. So we start at the beginning where we build relationships with patients within an indication. We do multi-omic experiments. Specifically, we're focusing on single-cell RNA sequencing. I will explain what that means. And then using this platform, we're generating a very rich atlas that is powered by AI. So, our patient relationships is what really enables us to scale our studies. So in the beginning, we build trust at the recruitment. As we start the study, in this case is going to be autoimmune flare study that I will be talking about. We're very transparent with patients about how their data is being used, what is being generated. We work with patient influencers and communities. And as the study goes on, we're constantly checking in with patients and getting feedback from them. And when the study is done, we continue that relationship with the patient. We give them their data back, we explain what it means, and we continue to engage with them. And that's really the key for us to be able to build up our patient network and recruit again. And so we're collecting small volumes of blood. And I mean, you guys are familiar with this. This is a PCSIG. And one question that we always often get asked is, okay, what can I get from capillary blood? We typically collect between 100 to 300 microliters. We get about 100,000 cells per 100 microliters. And we're also able to get plasma. Devices like Tasso and Red Drop give you a little bit more on the plasma side. So in terms of what you can measure from capillary blood, we're specifically focusing on single-cell RNA sequencing, but you can also get genetics, immune repertoires, and cytokine profiling. Um, I don't know my audience here, so I'm going to explain why we're focusing on RNA and talk a little bit about single-cell RNA sequencing. So why focus on measuring RNA? As you know, the dogma goes, we start with the DNA, which is the blueprint from which the instructions get made, which is the RNA, and then we have the end protein. The reason we're focusing on RNA is because we want to understand, because that is the readout that tells us what is changing over time and in response to our environment. Our genes are not everything, as we know. And the RNA is specific to each cell type and each tissue, and it has a different state. It is kind of the emergence of who you are. And it is, with small volumes, it is easier to amplify it and sequence it compared to proteins. We use a provider for single-cell RNA sequencing called 10x Genomics. If anyone ever has questions about single cell for me, I'm happy to answer. We tried a lot of the other different platforms. So what happens is a little bit of science here. When we extract the white blood cells from the capillary blood that the patients send us, it goes through this machine, which encapsulates each cell into this droplet, and within a droplet is a chemical reaction. So the cell has RNA free floating here, and there's a bead that has these little anchors that capture the RNA. So the reaction proceeds when the cell gets lysed and all the RNA gets dumped out of the cell, and this bead, which has capture tags that captures the polyadenylated side of the RNA, captures the RNA. So we're able to capture many different RNAs in an unbiased fashion. So the next thing that we do is we do this method called multiplexing, where we combine different cells together. So we basically label who did the sample come from and what person did it come from, and that's what really lets us keep our costs down and allows us to do these high throughput experiments across many people. With this platform, we were able to build a large immune atlas that is highly differentiated. So we've done this across time and with many different diseases. And at each time point, we're capturing relevant metadata, such as what therapies the person taking, what lifestyle they have, any comorbidities they might have as well. And this atlas is for each study that we do. By having this reference atlas, we're able to compare it to healthy people, to other diseases. So that is kind of the ecosystem that we're trying to build, and how we work with our partners. Okay, let's go into the actual study data. All right, so we have chosen to work with autoimmune diseases, specifically inflammatory bowel disease, for this study. And the reason being is because these diseases have a waxing and waning component to them, which means they flare up, they have disease activity, and they go back down. And this is something that a lot of people have to live with. In case of IBD, the prevalence is about 3.1 million in US. And there's also a lot of room for improvement in terms of medications. And this is one of the things that we're trying to focus on, is help understand why certain drugs work and others don't. So you see that the efficacy is, there's much to be desired there. All right, so let's go into how does going decentralized actually affect the data we're collecting. So we've done a longitudinal study where patients are collecting samples during flare events as well as their baselines. So in case of a flare, so every autoimmune disease has a different flare, right? In case of IBD, it means your stomach is hurting and there's usually blood in your stool. And we also use a Harvey-Bradshaw index as something that clinicians use to determine if somebody has a flare. In case of rheumatoid arthritis, which is the controls for our study, these folks have joint pains, and we use the metric there that clinicians use. So we basically took all those questions and we codified them into a survey. So we have 3 flares and 3 baselines, as I mentioned, and we recruited people based on the criteria, whether they have Crohn's or ulcerative colitis. And so far, we have screened 500 people, 74 we were able to enroll in just 9 months. And as you guys know, these studies are difficult to conduct. By going decentralized, we're able to go much faster. And since the person had their first recruitment call in 4 weeks, they're able to get their supplies at home. So now they have these kits, and whenever they're flaring, they can take a sample. And one of the things that we get when we pitch our company and what we're working on is there's this always kind of doubt about patients, right? Are they going to follow the rules? Are they going to be compliant? And what we've seen is that they are very compliant, and 88% of the samples that come into our lab are able to pass quality control. Another thing that's really awesome about going decentralized is that you are reaching people where they are. And I know that diversity is a big point in conducting these studies because drugs have seen poor outcomes in underrepresented populations. And without any targeted recruitment, we were able to match the ethnic distribution of the US Census, which was really cool. So we have 74 participants across the US collecting samples across flares and across baselines. So going decentralized did not just speed everything up, but it also made this study and made the science actually capture the profiles across diversity of people. Okay, what kind of new molecular targets can we detect during disease activity? So one of the things that we're doing is we're like every person, has 3 samples for flare and 3 samples for baseline. So we don't have to compare across people. We can compare within a person. So the self baseline is able to really reveal cell-specific signatures that differentiate patients. This is not something that you would be able to do in a cross-sectional study. So what we see here is that this is the x-axis is flare and y-axis is not flaring. We see here that this person has elevated IgM memory B cells during flares. And these folks have NK cell abundance elevated during flares. So this is something that's really cool that you can see when comparing within a person. Here's another, as I said, we're looking at cells as well as genes within those cells. So this is for CD4 memory T cells. What this shows is that this gene is something that you would not be able to see a difference if you were to just, take everyone's flare samples and take everyone's baselines. It would have no statistical significance. But when you actually compare a person's baseline to their flare, this biomarker comes out, and this is something that would have not been detected if we were to do things cross-sectionally. Another thing that I think is quite cool here is that you can see that this person's flare gene expression abundance is the same as this person's non-flaring. So this just shows that we're very different. It's important to look across time. This is just an example. So we have so far sequenced 24 of the people that completed the study. I'm just showing 5 of them here. This is just something that corroborates a finding that we saw in the literature. So this is an enzyme called CoQ10A, and it's been known in CD4 regulatory T cells to help basically downregulate inflammatory responses. And what's cool here is that we see a downregulation of it in flare. That means we don't know if it's either doing its job or the flare is downregulating its activity. But it's also been shown in a study when a doctor has supplemented this enzyme for UC patients with their existing therapies, that they were able to maintain remission. So it was really cool to see this effect in our study, matching something that we've seen in public literature. And I'm sure there will be a lot more of those. So if any of you are interested in talking about the data or working with us on the data, I invite you to connect with me. Okay, and can molecular signatures be used to stratify patients within an indication? So everyone knows this data, machine learning and AI is hot right now, and it's for a good reason. Biological data is growing quite big, and it's hard for one person to just analyze it. As I said, like when we're looking at this data, we're looking at a person's 10,000 cells by 30,000 genes, and we have many people and many samples. So what we've done is employ machine learning to help us understand, do people stratify in meaningful ways? So this is more of like a control, something that you'd expect to see. What we see is that the baseline samples within a person are more similar to each other than flare samples. And that makes sense. Flares have more variability than your baseline state. Another thing that we see here is unsupervised machine learning was able to identify clusters of participants with similar immune profiles. This is more of a control case where males cluster different from females. So this is just in CD8 T cells. And this makes sense because the gene expression is different. So here we also see that this group of patients has similarity in natural killer cells as well. So in our study, we were able to cluster patients better. Usually what machine learning does, it clusters samples more effectively than sometimes just a patient labeling. So here we see that this cluster of patients that have Crohn's disease clustered together. And what was interesting is that when we looked at differential expression between patients' flare versus their baseline using machine learning labels versus that of the human, we were able to see more signals. So, even though we use these kind of canonical ways to define disease, I think looking at things molecular will help us go even further to really achieve this idea of precision medicine. So this is now going to a completely different use case, exiting the autoimmune flare study. So what I've shared so far is a few of the findings from autoimmune flare study. I do want to talk about another study that we did with, actually directly with patients, called Remission Biome. So in this case, we're working with a disease called chronic fatigue syndrome. So we had a patient scientist, Tess, reach out to us, and this patient population had, they haven't seen any drugs, any treatments come out for them. So they've been really taking things into their own hands. So they've been trying out, their own interventions. So in this case, they wanted to measure their molecular profiles before, during, and after a perturbation. So we sent them the kits, and they took 3 samples for their baseline, and they did an intervention using a combination of antibiotics, and then we measured their new baseline. And what was interesting to see here is that we were able to detect cell-specific signatures before, during, and after. So this is just an example, and I don't, kind of a warning there. This signal could be attributed to other things going on in this person's life. This is more of a technology showcase. But what was interesting is that, we found something that was previously discovered in the literature, is that CD14 monocytes are acting differently in these patients, especially in their, in their worst state. So we see here at baseline, CCL4 is high. It's a known inflammatory cytokine. And then when they did a perturbation, they now, this gene was downregulated, and even weeks afterwards, it stayed at this new baseline. And this patient reported feeling much better after this intervention. So these are the kind of things that we're able to discover with our platform by going longitudinal and looking at diseases that have these waxing and waning symptoms. All right, so as I said, it'll be a short talk, and wanted to be engaging, want you guys to ask questions. So I will pause here. Stop sharing.
Thank you, Tatyana. That was a short presentation, but very impactful, very interesting. Thank you so much. So please, if you do have questions, please submit them by the chat function. We've got plenty of time here. We've got half an hour to talk about questions that people might have. So please submit them via the Q&A. We do have a question here from Cindy. Great presentation. Are you looking to integrate other types of omics? I'm working in the glycomics space to develop prognostic and predictive biomarkers for IBD.
Yeah, thank you, Cindy. We are looking to integrate other omics as well. So, as I said, right now we've been focusing on single-cell RNA sequencing. We can do genetics, and we're currently trying to do cytokine profiling using the Olink platform. If you're working in glycomics space, I would be happy to work with you and see what kind of data we can pull out from these small volumes. But we would like to really integrate, as much of the signal as we can.
Thank you, Tatyana. I have a question about the stability of the samples. Obviously, you're collecting these samples, self-collection, I guess, in people's home, and they're liquid samples, I'm guessing.
Yep.
Are there any issues with the stability of the samples, shipping them to your labs?
Yeah, so let me just show you what the kit looks like. I think it might kind of give you, okay, I'll just share again. Okay, so this is what the kit looks like that every patient gets that enrolls in our study. Right now, everything is on 24-hour timers. So everything we do is when the patient, okay, so what happens is a patient has a kit at home. When they get the kit, they take these 2 cold bricks and they put it in their freezer. So they're there whenever they're ready. So say a patient messages us and saying, hey, I am flaring. What we do is we talk to the patient, we schedule their pickup, we assign them a time to do the collection. And so this is the device that we use from Your BioHealth. There are other ones in the market, as I said, there's Tasso and Red Drop. And so when they're flaring, they take out their ice bricks, they take a sample, there's instruction, we monitor the temperature and they pack it into the kit. So everything has to be done within 24 hours in terms of the collection. So I'm just going to exit out of here so I can see you. Okay. Which is, it is still a little bit limiting. And, when I told you guys about the 88% of the samples coming back past quality control, a lot of it is because of actually FedEx, right? When it was cold, some of the samples got delayed. So we've shown that within 24 hours, the gene expression correlates a lot with that of the 0 hour. So that's why we do 24 hours. We are actually developing a preservative that lets us mix the blood with our preservative, and you can just ship everything at room temperature, no cold shipping. But that will probably be available in the next few years. Right now, we're just sticking to 24-hour shipment. Does that answer your question, Neil?
Yeah, absolutely. So does that limit the region that you're able to work with at the moment? I'm assuming it's you not working outside of the United States. Are you even working outside of the West Coast of the United States?
So we are working across the U.S. We are able to get shipments from the East Coast. A good chunk of our participants are also in the East Coast. We can't work in states like Alaska. We usually can't get the shipments within the 24 hours. So that's has been our biggest limitation. If we are working with partners, we can also teach them how to do our methods. So if they have a lab, we can kind of deploy where they are, and they will kind of have to, work our platform locally. But right now, yeah, we are focusing on US, but hope to expand.
Okay, thank you, Tatyana.
I see a
There's
question from Adam. Oh, I was
Yeah,
gonna read it. Yeah, Adam.
you go ahead,
Okay,
Tatyana.
okay. Adam's asking, can you clarify how you're using AI? Your presentation seems to indicate more machine learning based on data sets versus AI LLMs, which seem to have problem with hallucinations. Yeah, that's a good question. I'm not big into the AI hype myself. I'm more of an experimentalist, and I'm always skeptical. We are using machine learning. We're not using LLMs. But I say AI just because that's something people are used to. We're using a platform called SCVI, single cell variational autoencoder. But yeah, right now, like whenever we run things, I always try to use the simplest model and understand what we're seeing. We're not using LLMs, so we're not really running into the hallucination issue. And we also, like, one other thing that you can always do is double check. Like, for example, when we assign, like we get the data, right? And we need to say, okay, what is this cell? Is this a B cell? Is this a T cell? We make sure to actually go and double check everything to make sure that the markers make sense. So yeah, they still need a lot of supervision. I hope that answers your question, Adam.
I'm sure Adam can let us know via message if there's anything missing. I have another question, Tatyana, about the patient experience. Are you doing patient questionnaires here, and do you have any feedback? And is it possible to be able to share your patient feedback with the broader community? Because I think, for myself, I think one of the things that's going to help get us over the line in getting these patient-centric sampling technologies more widely accepted is that voice of the patient. It's yes, it's enabling you to hopefully do the science you want to do, but if it really is if you're getting good patient feedback, or it's helping with recruitment or things like that. I don't know whether you've got any feedback on those side of things, the patient engagement side of things, patient feedback.
For sure, we have a lot. We're learning a lot. So in terms of the patient experience, so what happens is we typically recruit in patient communities on Facebook through patient influencers. People sign up, and we do a screening call, which is usually 30 minutes to understand if they would be a good fit for the study. And if they are, then we send them kits and we check in with them, to see are they flaring. And one of the most important things we've done is educate our patients to be patient scientists, because a lot of the times, like in the current way things are done, patients are usually told what to do. They're usually told not to really think for themselves, and in this case we're kind of educating them. It's like, hey, when you're flaring, you want to take a sample. And patients are very smart. They, like, for example, if they we actually had a few of them get COVID, and they're like, hey, I probably shouldn't take a sample right now because that would interfere with my flare signal. Like, you want to know, is the signal coming from a flare or from the infection? So that patient education is really important. Um, we do surveys for every sample that they take. So we would like to build our own app for this, but right now we're using Survey Sparrow. So for every time they go and take a sample, it asks them questions. So it has the Harvey Bradshaw basically codified into the survey, and then we ask them other things. One of the biggest learnings we had was medication entry. So um, just because a patient has autoimmune disease does not mean that they're not dealing with a bunch of other stuff. So some of the burden that we've seen on the patient is entering their medications. Um, like I was on a call with one patient, and they're like, I'm not going to input my 20 medications into this. I just want to take the sample and send it off, right? So, just inputting the medication can be a burden on itself. So that was one important learning that we had. And also just understanding that sometimes they won't complete everything, that you will need to check in with them. Have that conversation. It's not just kind of like one and done. It's really important to have that back and forth. Like, if you see something in the survey that you as a researcher are confused about, we have access to the patients to confirm that, right? You don't have to go through layers of different kind of like CROs to verify something. So having that channel open has been important for us to learn.
And do you find, because you're working with patients with rare disease, that they're particularly motivated to taking part? Because obviously participation, sticking with the trial, actually collecting the samples at the right time, the right place, it is a big challenge. And
No, for sure. And everyone has different motivations. When we started our study, our incentive was going to be, hey, you're going to get your research data back, which is really new and novel. You get to see your different cells, your different genes, and we were able to get a good amount of folks signing up. But we noticed that it only recruits a certain type of person, and you have to realize that you have to provide different incentives to different people. So because you want to have a diverse data set. So then we changed our incentives to also include payments. So for every 3 samples that they complete, they get a gift card to Amazon. Um, so those things have been really important to integrate.
yeah, that's really cool. And some of these findings, above your scientific findings, just these patient behavior findings to home sampling, is there any way you can share that kind of data in the future?
Yeah, no, for sure. So I've only shared those 5 people, but we just finished sequencing 25, and there's 74 people enrolled. So it's going to be a pretty big study, and we already have a company that's actually working with us and that's very interested in the data set, and they're going to be developing their target program from this. So it's a very rich data set, and hopefully has a unique look into things we couldn't see before, especially since we're capturing the disease activity. I do want to mention one thing. In the future, we'll also be integrating the ability to get medical records from these people. So when we have the exit survey, we're figuring it out. Because one thing I learned is that it's really expensive and really burdensome to get medical records. And because we have this patient relationship, it's easier to just ask the patient to upload the records than it is to just go through like Datavant.
Yeah.
So it's another thing that we're also learning. Like, is it easier to get the patient to do it, or do you need to go through these services? I do want to mention that we are also working on other indications with with clinicians. This is our study where we're doing entirely on our own, just because we wanted to see how fast we can go when we're not part of the healthcare ecosystem.
One thing, so the PCSIG last week ran a workshop focusing on how we get this kind of sampling out there for multi-site pharmaceutical clinical trials. And one of the big discussion points was around, do we need to confirm that the person is who they say they are, that is collecting the sample? Is that a problem with rare disease patients? Is it not something you need to be concerned about?
I mean, that is an important point, right? It is something to worry about, because especially if you're giving out incentives that are monetary, you might get that. But we do a lot of screening in terms of talking to the person. I think some of them we've done it on video, and usually ask questions that are very disease specific. So I personally haven't run into someone where I think, okay, they're being shady, they're lying that they have this disease, just because a lot of this nomenclature is so specific and so like you experience it. I haven't seen it, but I can imagine that being a problem at larger scale. So
Yeah.
you probably do want to have some sort of verification.
And there was very much a difference in opinion of what should be done, going from it's just too expensive to monitor everyone that they're collecting the correct sample, and we've got to trust people, through to no, we need some other form of confirmation if our data is going to mean something. So
You're going to see it in the data as well. If a person says they have a certain disease and they don't, and they're an outlier, right? And you had this gut feeling that there's something wrong there. You're going to see it, right? The data will reveal itself as well.
Yeah. So it looks like we're not getting any other questions coming in, Tatyana. Just give everyone a couple of minutes to submit any last minute questions. Are there any other comments? Anything else urgent that's on your mind that's occurred to you during the last 35 minutes that you'd like to speak about a bit more?
No, not that I can think of right now, but as we do the study, we're sharing this constantly. We're making sure we're sharing these things with patients as well. So, but if anyone has any study ideas that they have that they want to dig into, or want to look at the data set that we're developing, I'd be happy to chat with them. Or if you have any other assays that we can try with small volumes of capillary blood, please let us know. We're looking to grow the multi-omics aspect. Can you share your emails? Yeah, I can share my email just in this public chat.
Yeah, if you do that, I'll just so I've got a few things I just want to present on to wrap things. Oh, here we go.
Great presentation. Have you looked at reproducibility of collection/results? For example, collecting 3 samples across a day and assessing performance and consistency. Yeah, that's a good question, Evgeny. So right now, when patients collect, so when we started the company, the seminal work is we actually did, as a proof of concept, we collected during the morning and during the evening just to see the diurnal changes. And there's a lot, right? Between the morning and night, the immune system is on a diurnal cycle. So when we do have folks collect, they typically collect at the same time. We ask to make sure that they collect at the same time. We're going to be able to see this as we finish up the study. But in the future, we would like to do a study that actually characterizes this aspect. So, when we look at the data sets in the future, we know when we see a change that their variable could also be attributed to diurnality and not to the disease. We'll be able to build stronger models around that as the data set grows.
Great
But
answer.
if you want to read more about it, I'm happy to share the original work that we've done, so you can kind of learn more about it. Let me just paste it here, actually.
Okay, cool. I'll give you a moment to do that.
Yep. Yeah, this is the original work that started the company.
Okay, thank you for sharing that. So as I said at the beginning,
I just want one more question from Evgeny.
okay.
Thank you for the answer. Is 300 microliters sufficient for all the analytes you're assessing, or are you looking for larger volumes of blood? It is sufficient for doing single cell RNA sequencing, because when we get the sample into our lab, what we do is we isolate the white blood cells using the stem cell kit. And for single cell RNA sequencing, we're typically loading between 5 to 10,000 cells per sample. So we're getting 100,000 or more, and we're only loading 10,000. So it's more than enough for us. We actually have extra vials. We are assessing the cytokine profiling. I'm a little weary about doing proteins, right? This is the whole Theranos thing where, it was promised that it could do a lot more than it can. So we are assessing other modalities, but for genetics, for single cell RNA sequencing, and so for some of the cytokine profiling, it's enough.
I thought we might get through without using the Theranos word.
Yeah, no, it comes up. Even surprisingly, somebody was comparing Grail to Theranos. So even though it's a venous blood draw, and another answer there, 300 micro. A lot of people do bleed more than that, right? It's basically how long are you leaving it on there, and yeah, we use tab because it's quite painless, but some of the other devices, because they have a sharper lancet, they're able to get a little bit more. So
Cool. So this webinar is being recorded and will be available for streaming quite soon. It'll go up on our website and on the evidence-based website as well, so people will be able to interact with it there. Just let me share my screen to wrap things up a little bit. If questions come up in the meantime, we can go back to that. There's not a problem at all. So just next month on the 17th of July, we have a couple of presenters talking about the acceptability and validity of microsamplings for serosurveillance in Kenya. So really looking forward to that, moving beyond the North America, Europe sphere, which is where we've been concentrating a lot of our work so far. So really looking forward to what Makobo and Rose have to say next month. So please take a look at the PCSIG website or the evidence-based website, and you'll find the links to be able to register for that. And obviously it will be available for free to stream after the event. And for those of you that aren't already sampling interest group, it's free to join. We're always seeking sponsors, but please take a look at our website. If you want to join our LinkedIn group, we're over 400 members now. Please feel free to join that LinkedIn group, ask questions, post your publications, tell people about your conference, things like that. Just don't be overtly commercial on there. But we're a friendly group, and the more members we have, the more activities we can get done. And the idea is to educate, to help people understand what patient-centric sampling is, realize the benefits of various different uses. And so if anyone's got any ideas for activities, please join our group and talk about it. And lastly, I'd like to thank again all of our partners and our sponsors, without which we wouldn't be able to do these webinars and share all this wonderful information with you all. And with that, Tatyana, I'd like to thank you very much for your presentation today. It was really super interesting. We had lots of time for questions. Thank you to all the viewers for your questions. You've got Tatyana's email address and the link to her information. So please feel free to interact with Tatyana. Feel free to interact with the LinkedIn group. And thank you all for your attention.
thank you everyone for joining and listening in. I really appreciate it.
Cheers all. Thank you, Tatyana.